introduction of DNA into the non-bacterial cells are called animal cells or plant cells. Okay. Various methods exist to transfer the DNA into the host, namely Electroporation, Microinjection, and partial bombardment or gene gun method. The gene gun method, very famous method, now recently used. So the various types of gene transfer method to be employed in the yeast, fungi, animals, and plants. Why? Because we have some specialized applications. The yeast, we need some specialized enzyme production. The fungi, we need some create the pesticides, insecticides. Animals, we need for foods. Plants, we need for growing our economy. So we introduced large number of genes into our system. Okay. Moreover, molecular biologists are forgotten the transformation technique. Why? Because it has a lot of demerits. Yes. So today, the improved technology was needed to instead of to face these problems in the transformation. So a lot of techniques are needed. So the scientists to jump into sophisticated methods for gene transfer into the host. Likewise, here sophisticated method means microinjection, electroporation, and back gene gun method. Yes. But sometimes the famous laboratory or famous scientists are employed the transformation method using Saccharomyces cerevisiae. Before the transformation, the yeast cells are treated with the lithium chloride or lithium acetate instead of calcium chloride, ice cold calcium chloride. So the lithium chloride and lithium acetate to play the role carry the DNA from the environment to the host Saccharomyces cerevisiae. Okay. This old techniques are nowadays are also used. So that's why I mentioned here. And also instead of calcium chloride, the lithium chloride have more features because easily binds to DNA, easily not degraded, easily ionized, and easily reacted to the buffer, not reacted to the DNA, and not reacted to the protein, and easily can sedimented. So if you are using lithium chloride, you can easily estimate how much amount of the external DNA going to involve the transformation. Simply called transformation efficiency, using simply sucrose gradient centrifugation and also density gradient centrifugation, we can easily measure. That's why the people are doing the old method today also. Okay, anyhow, sophisticated methods are needed for efficient gene transfer to the host. Okay. Now, transformation of the gene or recombinant molecule into the individual cell. So the previous transformation, conjugation, transductions are transfer the DNA in number of cells. At the same time, the transfer of DNA into the number of cells. So this is a very drawback So the transformation of DNA into the single cell, we can easily isolate it and manipulate. So these sophisticated techniques are used to transfer the gene and transfer the recombinant DNA only in the single cell. Single cell called a single protoplast cell or plant cell, single embryo, a single zygote, a single stem cells, a germ cells. So whatever we want to insert single cell. For example, if we are treated the genetic disorder through by the gene, we can transfer the DNA into the stem cells. Suppose we can produce the recombinant plant, the recombinant DNA injected into the single protoplastic cells. Protoplast means removal of cell wall, the plant cell. It's called protoplast. So even leaf cells, stem cells, root cells, epimeristem cells. So whatever cells we can use. Similarly, we can transfer the gene into the embryo during the in vitro fertilization. It's called IVF. So a lot of applications are there, but the transformation only carried the individual cells. Before studying the technique, number of DNA molecules, recombinant molecules transfer into number of cells at the same time. Okay. So why that old transformation principle not obey here? The important parameters there, because animal cells or plant cells have some barriers. Because if you are taking the human cells or animal cells, it's fully covered with the collagen, fully covered with the myosin. So number of protected proteins are there. So we cannot easily introduce the DNA into the cell. That's why the scientists or researchers are transfer the DNA into single cell. First we can isolate it and separate it and treat it and finally produce a single cell and easily transfer into DNA. Once again transferred, we can culture and regenerate in the new plants. But animals we cannot degenerate the single cells. Suppose we transfer into the single cells in the embryo, we can reimplant into the uterus. So once again we produce the well-developed recombinant organism. So main barrier is there. So this characteristics to play a role. These transformation techniques employed in the single cell. Okay. The cultured animal cells usually lack of cell walls. So after transformation, the cell contains no cell wall. So without cell wall, the cell not easily to culture. So this is one more additional point. The transformation technique employed in the single cell. Okay. So easily not transformed. The DNA is precipitated on the cell surface using calcium chloride, even calcium phosphate, or the same principle is liposome based delivery of DNA. So the liposome based DNA delivery and the calcium chloride transformation we cannot do. These techniques are not do in the plant cells, animal cells. Why? Because it's a lack of cell wall. Yes, but we cannot easily degenerate it. Kept your mind? Yes. So don't forget. Okay. There are some techniques are available to destroy the cell wall and produce the protoplast or zygote. Yes. So, we are already learned the last time transformation of DNA to the yeast. So we can produce the saproplast using antibiotic based saproplast generation or enzyme based saproplast generation. So by these enzymes treated with the corresponding cells before the cell pad up of cell wall. And also why we do this like means just to compare the transformation. If we transfer the DNA into the bacteria, we have competent cells. Similarly, in here we introduce the recombinant DNA into the either plant cell or animal cell. First we convert it into protoplast, either plant cell converted to protoplast, animal cells converted into protoplast. So the protoplast equal to competence. Then only the DNA could be transferred by the electroporation or microinjection. Okay. So after the protoplast, we can generally employ and quickly, readily insert the DNA into the host. Here, very simple explanation mentioned here. This is a basic transformation technique used in animal culture. So this is animal protoplast culture, and this is recombinant DNA, and also treated with the calcium phosphate. The calcium phosphate binds to the DNA. The mechanism of salt out, the recombinant DNA get precipitated and goes to bind the surface of the plasma membrane. Maybe get inserted. The inserted fragment further can proceed. Replication, transcription. Okay, this is an old method, but it has some demerits. And why the people goes to sophisticated gene transfer technique. This is animal cell, considered as animal cell. Animal cells means this is a treated animal cell. This is protoplastic animal cell, and also fused with the liposome based DNA. This circle forms liposome. The liposome contains DNA, so it can easily fuse with the plasma membrane. So through by the endocytosis, the DNA goes to inside and finally enter into the host chromosome, whether the recombinant DNA have recombination ability. Okay. Moreover, the plant's protoplasts is a normal plant cell. So remove the cell wall, degrade the cell wall using by enzyme or antibiotics. The cells converted to this cell. Losing the cell wall or degrade the cell wall, converted to protoplasm. This is a protoplastic cell or competent cell, equal to competent cell. Here is a competent cell of plant. This is a nucleus and vacuoles. Vacuoles, all the plant cells have vacuoles. Why? Because the vacuoles used storage function, containing our nutrients, foods in the plants. If you are taking the potato, 97% of the potato cell occupied by the vacuole, remaining 5 or 6% of the part of the cells occupied by the other components. If you go to search the Google, you know very well about it. Okay. So nucleus and vacuole, plant cell, convert to protoplast. So foreign DNA or recombinant DNA may be treated. So after treated, it was selected. The selected recombinant cells once again degenerated into old cell or parent cell. And also the plant cells are easily degenerated, but animal cells not easily generated. It some medicinal techniques are needed. Okay. Finally, we produce transformed or recombinant plant, a genetically modified plant. Yes. So you know the basic difference between the this is a DNA precipitation, this is a liposome based DNA delivery, this is a microinjection method. Yesterday we learned a lot of points electroporation. Once again in reminding, electroporation is a technique to employ the transfer of gene to plant and animal cell. Yes. Transition formation occur when means if the protoplastic cell subjected into the electroporator at the time of electric field conduction. So at the time of electric field application in the protoplastic cell to feel transition formation, that means small modification. Yesterday told small modification is happening. The transition formation of pores to be created in the cell membrane. So at the time the environmental DNA may get to insert through by the rate of amount of electric field applied, E, amount of E applied. Yes. Okay. The protoplast was generated by corresponding degenerative enzymes. So apart from the electroporation, two methods are there. One is microinjection. Next one is called biolistics. Biolistics means otherwise called gene gun method, or otherwise called partial gun, or sometimes called partial bombardment. All the terminology are same. Biolistics, partial gun, gene gun, partial bombardment, everything is the same. And one more technique is microinjection. The highly advanced physical methods are available to introducing the DNA into the cell. First one is microinjection. The microinjection is very interesting technique. This technique effectively are widely used for transfer of gene into embryo or zygote. Embryo and zygote. Sometimes maybe played in the transfer of gene into plant cells also, but mostly used in the transfer of gene to embryo. Here is fine needle. Here is simply denoted as a syringe. So the needle will not look like micro syringe are there. So using the syringe, the DNA solution containing recombinant DNA and also cytoplasm solution, hypotonic solution is there. So using the scanning electron microscope, the cells placed on the scanning electron microscope. So using the needle, we can inject the DNA solution into the single zygote. This is called IVF, in vitro fertilization methods are doing this method. Okay. Second method is biolistics or partial bombardment. It's very interesting method. The method involves bombarding the cells. Bombarding cells means just which cell we want to transfer, the host cells placed in the grid structure. That means placed in the plate-like structure. After placing the host cell into the plate, the high velocity microprojectile. So you look like this. So this is a plate. This is a plate. I told. This is a plate. Here, this is our host cell. This is our host cell. Using some adhesives. So after here, this look like a piston. You know the piston. This look like a piston. So here our gene of interest, recombinant DNA molecule placed here. Using the piston, this piston is not a high velocity piston. High velocity piston immediately strike into this section. Immediately the recombinant molecule bombard to the cells. Bombard means you strike to the cells and very high speed. So that time the DNA molecule get inserted into the cell. So the target cells are bombarded with the microinject. Microinject means our recombinant DNA placed in the small granules, a small DNA-like structure. It is called the microprojectile. So the microprojectile is inject into the host cell. Immediately the DNA goes to inside. So this firing pin, high velocity piston. This is microprojectile recombinant DNA. Yes. Second method. Usually the particles of gold and tungsten that have been coated with the DNA. The microprojectile means, so the DNA attached with the DNA attached with a gold particle or tungsten particle attached with the tungsten particle using by some method is that using alkaline phosphatase we can remove the phosphate group from the end and easily add to the gold nanoparticles or phosphate or isotope, whatever we want to be added. So just like the attached the gold particle into the DNA, it's called a microprojectile. So the microprojectile placed here and the piston is working and immediately deliver to here the DNA and may get inserted. The microprojectile are fired at the cell using porcelain. The technique is called biolistics. So it can used for transfer the DNA into several different types of cells because the transformation efficiency may be high because these are very sophisticated or gene gun method, just like a gun. Okay, this is an image I mentioned here. This is a potato cell. This is a single potato cell. Microinjection method transfer the DNA. The protoplast cell. The protoplast derived from potato cell. So using the glass capillary tube. This is a glass capillary tube. This tube used for holding the single cell. This entire setup placed in the some scanning electron microscopy. So using the capillary tube to holding the single cell because we are injecting DNA solution into the cells. Okay. By this gentle suction. So capillary means using suction. The gentle suction, the cell is holded here. So don't move for the injection purpose. So the microinjection needle made heated the glass capillary. So fine point is the very fine point up to nucleus. The fine needle goes to up to the nucleus. Yes. The very fine needle. The fine needle used to insert the DNA into the cells. So this is a famous photography shown here. Okay. Next. So that these are the two techniques are there. Sometimes the transformation of whole organism. Suppose we cannot take the DNA because we cannot separate the DNA. So that time the very crude method is called whole organism transformation. This transformation, whole cell transformation, only takes place in the plants, not animal, not in the bacteria. So the monocotyledon, especially monocotyledon species, monocotyledon means single cotyledon species. Example: rice, wheat, corn. So this like cereals or pulses. So here we want to breeding the plants. Usually we are taking the stem and join together and place the grafting. So this is one kind of breeding, but the internal breeding, suppose we want, means we go to transfer the whole cell into the cell, whole cell into the another host cell. So that time the entire chromosomes are recombinated. So finally produce the new hybrid, especially in the monocotyledons. So that's why the rice varieties are produced by this method. Especially cereals are grasses. Yes. But the whole cell transformation not done in the animal cells because here some limitations are there. Maybe the whole cell transformation takes place in the fertilized eggs. Fertilized eggs. Suppose some broiled chicken or broiler fish, or broiled goat, a broiler rabbit. So number of hybrids are there. So those hybrids are maybe hybrid breeding into this technique. Okay. So fertilize the egg, one egg fertilized to another egg. So finally we hybrid egg get produced. That is also called transformation, but not transformation of genome or recombinant DNA. The whole cell can be transformed. This transformation, the microinjection, effectively employed. So here, the concept map displayed, so because you easily understand. Gene cloning requires host cell. Host cell means either prokaryotic or eukaryotic, such as E. coli and Bacillus subtilis. We are using only E. coli. And also, if you are taking the eukaryotic, Saccharomyces or plants or animals. So need the method for introduction of cloned DNA, such as transformation, transduction, transfection, or packing the in vitro method, microinjection, biolistics. These are the method of using the transformation of gene into or introduction of gene into host cell. Yes, gene cloning requires host cell, prokaryotic, eukaryotic, E. coli, Saccharomyces. So introduction of gene into transformation, transfection, packing of in vitro. So these tools are used by M13 and T4 bacteriophage. Microinjection, biolistics. Bombarding. Okay, come to here. Cloning use as a vector. Vector a carry molecule. Carry molecule first one which should have or many have. Should have origin of replication, yes, essential. Cell markers essential. Restriction size essential. So without these characteristics, we are not considered as a vector. Okay. And can be inserted into host cell by these transformation method. Yes. Okay. Maybe have, we should have, maybe have multiple cloning set or expression set. In the case of expression vector, schedule vector, the multiple cloning sets are needed. Similarly, multiple selectable markers are needed when it's effectively selected. Okay. So which is should have air magnet. Okay. Then two main types. One is plasmid, one is phage. M30, T4 bacteria. Plasmid, PUC18, PBr322. Number of plasmids are exist, which are circular molecule. Both are circular molecule. So and have been specially constructed. Yes, both are specially constructed. If lambda phage or M30, yes, two types are there. Here, insertion sometimes it is called. So simply defined here. When you are taking the lambda DNA here, suppose we insert the foreign DNA into that. Okay. Suppose we inserted the foreign DNA into the lambda genome, just inserted into the genome, so the genome size is increased. This vector is called insertion vector. Okay. Similarly, the lambda genome is there, the foreign DNA is there. We delete some segment. Similar like segments are deleted here, deleted to replace the deleted segment using the recombinant DNA. It is called replacement vector. So both are employed in the lambda phage, such as example lambda GT10 we all know, and charan 68. This is a insertion vector. EMPL4 and charan 40. Charan 40 means insertion or replacement vector of lambda phage promotion. Okay. Other types are there. We know cosmids, coside, phage mids like this. These are the phage mids. Yeast artificial chromosome and some viruses. Okay, you know very clear about that. Yes, that's all about. Please unmute your voice.